Hey, what's up, Hognose fans? Thanks for tuning in to Hognose One. So another talk, yet again, genetics. Somebody said, Kevin, how in the world did you get interested in genetics? And it takes me back to the story you've all heard about hopping into the pet shop over at Richard Evans' place. My parents would say, what do you want to do today? It's Saturday. I said, I want to go to Pets Plus and to look through the big display windows. And you couldn't get to where they had all the aquariums and little, critter keepers set up with everything. But you could see those through a large plate glass window that was probably like 4 by 6. You just look in there. He'd have little handwritten notes outside, all reptiles 20% off, or hognose snakes today, $19.99, or whatever the case was. But I always was drawn to the case of hognose snakes that was there, the 10-gallon tank. And one of the employees one time said, you like hognose snakes? And I was like, yeah, they're so cool. And they said, you need to talk to Richard because he just got some professional pictures done of an albino that he got in. And I was just floored because I'd only ever seen an albino Burmese. That was the only albino that I was familiar with. And so I went down the long, dark hall, knocked on his door timidly, and he answered and showed the little picture of the pink pastel. And I was, from then on, I was snagged. I was like, I have to know more about this. And he told me about another snake he had hatched out from a wild-caught to wild-caught pairing called a hypo. That's what he was calling it. He said, it's just such a light animal. And so that began my journey. So this has resurfaced in recent years. 4 years ago, I reapproached him, I think around COVID time, when I was doing the traits series because I wanted some viable pictures. And he found, miraculously, they weren't destroyed or lost after 20-something years. He found the original little slides that had pictures on them of the very first hypo, which we know now as Evans hypo. He found pictures of what I believe to be the very first frost. I know a lot of times we've attributed other people with hatching out the first caramel, but I think they all probably originated here and we just didn't know what we were looking at. So look at some of those pictures today and you just kind of get your assessment. And again, I don't intend to say we should visually look at every single thing to try to make determinations. We should, at least for now, attempt to do test crosses. With recessive traits, we need to breed them back, breed them to outcross bloodlines, meaning something they're not related to hopefully hone in on and prove what certain lineages are. Okay, we're at the verge or on the cusp of this genetic research that a hognose genome has been mapped, and what that means is just a digital database of all the genes that a hognose snake genetic makeup, the genome we call it, has for expression, for form, for function. And they build on that by the amount of different DNA that's submitted. You can submit DNA via blood, you can submit DNA via shed skin, and other methods. But it's really amazing where we're getting to. And that will really help to eliminate a lot of the stuff long term, like a lot of the questions we've got about where these genes originated or what genes, how they interact with one another. Are they really dominant? Are they just, polygenes? Are they recessive? Just the different types and the inner workings or inner relationship that they have when they come out. A lot of cool research is being done right now. I talked to a friend recently that's doing a study, awesome stuff, just to figure out and hopefully help us to label some of these genetics. Cool stuff. I'm really excited where the hobby's going. Hopefully these pictures I'm sharing with you today will be of benefit. Thank you to those that have submitted these, and thanks to Richard Evans, another photographer, Michael Price. And as always, guys, thank you for the like and subscribe, and I hope to catch you on the flip side.